Hello, and thank you for joining me today on this presentation about research and development, particularly in the laboratory context. So what we'll be looking at is the critical assessment and analysis of published work and how that is done, and also using an evidence-based approach within the laboratory context and what that means and why it is important to know how to do that. I'm Dr. Janine Martins from the Faculty of Health Sciences at the University of Pretoria in South Africa, and I'm affiliated with the departments of Chemical Pathology and Public Health. So in this presentation, we will look at four main learning outcomes, and first of all, it is to understand what evidence-based medicine is, its definition, and then looking at the introduction to critical appraisal and how that actually is done and the role within EBLM or evidence-based medicine. Then looking at evidence-based approaches in the laboratory medicine setting and how this differs to normal evidence-based medicine. And then finally, to try and pull it all together to understand what is R&D, research and development in the laboratory context, and how this relates to new markers, the development of new markers, and also what kind of jobs and roles can we have as chempaths and laboratory personnel in this area of chemical pathology? What is evidence-based medicine? According to the National Cancer Institute, it is a systematic approach to medicine. And that is key, a systematic approach in which doctors or health personnel use the best available scientific evidence that is available at the time. As we know, science is dynamic and always changing, and therefore it is about the best available scientific evidence at the time that's based on clinical research, and it assists in making decisions, particularly clinical decisions, and about the care of individual patients. So ultimately ensuring the best outcomes and safety of treatment of our patients. Evidence-based medicine involves the critical appraisal of research, a detailed assessment of bias, and as we can imagine, there are always elements of bias that we have to look out for. There is a risk of that in research as well, even in the best conducted studies. And then finally, critical reading of methods and results of research publications. So critical appraisal of research is carried out to reveal if the methods and results that we used valid enough to produce useful information. And this gives us a guideline for appraising medical research. So just because something is published doesn't necessarily mean that it meets the criteria for a valid study or a study that's good enough that we can use in practice. Firstly, we should review a structured abstract, or there should be a structured abstract available. And then we need to look at the key objectives of the research. First of all, what are the authors trying to measure? Secondly, what are the objectives? Can they be easily identified? And finally, have the research objectives been met? So it doesn't help having an objective, but they're not actually measuring or having an outcome based on that objective. So according to the Center for Evidence-Based Medicine, which is a very good resource to always refer to when you are doing any work on evidence-based medicine, process or the concept of critical appraisal is the systematic evaluation of clinical research papers, and it aims to answer four specific questions, which we will look at in a minute. Critical appraisal also includes addressing the risk of bias, which we've alluded to, and that involves a careful reading of methods and of the results. What are all these four specific questions? And these are referred to as the critical appraisal tools, which can be found on the website of the Center for Evidence-Based Medicine and are key in any research paper that we look at. First of all, did the study address a clearly focused question? So when we do research or when our colleagues do research, there should always be a question, a hypothesis. Secondly, did the study use valid methods to address the question? So did the methods that they use appropriately match the question that was asked. Thirdly, are the valid results of the study important? So results might be valid, but are they actually clinically important? And finally, the fourth question that should be answered is, are the valid results applicable to my patient or population that I'm trying to apply these results to? We have to remember that results published in another area, field, or country or population might differ to the one that you're actually working in, the setting that you're working in. And those differences have to be taken into context. It might not be a situation where you can immediately apply it to your own setting. So looking at the process of a detailed critical appraisal of a journal article, we have to carefully and systematically examine the research. It is always about the research trustworthiness, its value and relevance to the particular context that we've just described. This is a very important element of evidence-based medicine. And there are 5 steps involved in terms of critical appraisal. We have to ask answerable questions. As we mentioned, formulate the right question. Formulate it in a way that you can actually interrogate it by using medical literature, and also in a way that you can find answers to the question. So there's always the sentiment of, if you've asked the right question, you will find the right answer. There are several tools to do this. So once you've identified a clinical problem, you have to define the question. And one of the main tools that are used in evidence-based medicine and critical appraisal in particular is something called the PICO tool. And I'm going to look at that a bit later on. This helps us to break down the query into particular components. Then we have to search for the evidence. So this is finding the evidence, finding the answer to our question. Often we can find pre-appraised resources, which will be very helpful. Then once you've found the resources, you have to critically appraise these results. And then, based on this critical appraisal, you decide what action to take from your findings, and you evaluate your new or amended practice based on these findings. And as we know, science is dynamic, medicine is dynamic, and we've definitely seen this with our current context in terms of the pandemic. We've experienced how literature was constantly changing, or new information that was released, and we've seen this in the history of medicine, how findings are always evolving and new literature is added. So to break this down easily and to give you a checklist when you are looking at a paper, you can print this out and keep this next to you when you are looking at research. It's almost a checklist that you can tick as you go through when you look at any research. And I'm going to just recap this. So do we have an important question or an important scientific question with an aim? Secondly, will the study outcome provide clinical significance to the reader? Does it have any relevance? It might be interesting, but it might not have any relevance to the clinical setting. Is the methodology well designed? And again, this goes back to, does the method that the researcher used and identified actually match the question that's being asked? Is it relevant to the question? And there are different designs, study designs that can be used, which we will also go over. Next, is the primary outcome variable properly evaluated? So did they use the right tools to evaluate the primary outcome, and are they reporting on the right things? They usually are primary outcomes to the study and secondary outcomes, and researchers always have to report on their primary outcomes and not only focus on the secondary outcomes when they are reporting study outcomes. Next, you have to ask whether the sample size is large enough to find a reliable and meaningful result. This is the difference between a case study, which is based on an N number of one patient, and where you want to extrapolate this to a larger population. If you have 100 results showing the same things, you start to have a pattern and you can deduce wider things that are more widely applicable to a population. Finally, you have to see whether there is a significant reason to doubt the validity of the findings. So this would be facts such as poor methodology, insufficient sample size, or other confounders. What are the modes of research or methodology that we've spoken about that you can look at, that you need to look out for? This goes back to the question, what is the overall study design of the study you are assessing or critically appraising? And there are different types. We have case report, we have case series, we have cross-sectional studies, cohort studies, case control studies, and also controlled trials. And these could be randomized controlled trials and other versions. So for many people who haven't been exposed to methodology, this becomes confusing. So case report is a study or description of one unusual case that you may have identified in practice. The case series is what the word says. It's a series, a number of studies where no specific hypothesis is verified and results from different papers are not compared. So it's just reporting the outcome of various studies or cases. A cross-sectional study assesses the frequency of characteristics in a defined population at a particular time. And a cohort study is an observation of a group of people with a disease that are monitored over time to detect changes. A case control study is where a group of people with the disease is compared to a control group without disease. And then our control trials, which is an experimental study where an intervention is applied to one group and compared to a group in which the intervention is not applied, or otherwise your placebo group. And these can have variations as well in terms of how they are designed and how they are presented. For example, whether they're randomized or blinded or double-blinded, placebo-controlled variations of them. And this needs to be taken into consideration also whether the designs that have been chosen actually match the question that's being asked. We haven't yet covered qualitative designs, which we will look at a bit later on in terms of how these fit in with these main overall study designs. So this brings me to the hierarchy of evidence, or what we have traditionally looked at in terms of assessing the hierarchy of trials, research papers, when assessing their importance. And these were tiered from level 1, systematic reviews, down to level 8, which included opinion of authorities or expert committees. Level 2 included well-designed randomized controlled trials. Level 3 included controlled trials without randomization or quasi-experimental studies. Level 4 included single non-experimental studies. Level 5 included systematic reviews of descriptive and qualitative studies. And level 6 were single descriptive or qualitative studies. And then, as we mentioned, level 7 was then about expert opinions and colleagues' input in terms of committees, reports, and other authority reports that were released. The hierarchy of evidence was then used to set up a grading system. So in the early 2000s, the grading of recommendations, assessment, development and evaluation, or GRADE, the working group was developed, and they developed a framework in which the certainty in evidence was based on numerous factors. So these factors were not based solely on the study design, which challenged the pyramid concept. Study design alone appeared to be insufficient, and it was a surrogate for risk of bias. Because there are certain methodological limitations of a study, such as imprecision, inconsistency, and indirectness. And these factors are independent from study design. They can also affect the quality of evidence derived from any study design. So just because you have randomized controlled trials and a systematic review was performed, if there was any form of bias in those randomized controlled trials, of course, then by default, your systematic review or meta-analysis would have certain limitations and would not necessarily be at the top of the pyramid. So the grade system therefore challenged the concept of purely basing the hierarchy on study design. And as we can see on the right, there were certain factors that were included that determined the quality of evidence, whether it was high, moderate, low, or very low. Study design did play a role, but was influenced by the study limitations, and also the scoring could either increase or decrease according to the quality factors that were identified. So evidence-based medicine progressed to recognize the limitations of evidence alone and has increasingly stressed the need to combine critical appraisal of evidence with patients' values and preferences through shared decision-making. And the way this could have been applied is an example here in our flowchart where we have the initial certainty of evidence. We have factors that determine the certainty of evidence, and they can either increase the value of the study or the validity of the findings, or it can increase it, such as increasing factors included the large magnitude of an effect of a study, dose-response gradient, or the effect of plausible residual confounding factors. Factors that reduced the certainty of evidence included risk of bias, inconsistency, imprecision, and publication bias. And then all of this led to the final certainty of evidence, where you would have a result of either a very high, moderate, low, or very low certainty of evidence. And this then led to the grading of clinical evidence. So as we know, generally results derived from clinical trials start with a high level of initial evidence certainty, and those derived from observational studies with a low level of certainty, although exceptionally this may vary. The grade of recommendation was then based from A through to C and D, and these had further subcategories such as 1A, B, C, 2A and B, C, 3A and B, 4 and 5, and it referred to the varying structure of the studies. The lowest grading was expert opinion without explicit critical appraisal, or bench research, for example. The hierarchy of evidence begins at the foundation with unfiltered information and evidence. It still does. The top of the pyramid represents meta-analyses, as we've mentioned, that are highly filtered and verified. So the question is, if we are using this system plus the grading system, where do we find the corresponding databases for these types of trials? So this diagram depicts some of the sources, which has either filtered information or unfiltered information, which divides the pyramid further into two sections. So for example, the source of meta-analysis that's considered filtered is using Cochrane Database of Systematic Reviews. All critically appraised topics could be sourced from the database of abstracts from reviews of effectiveness, otherwise known as DARE, or from the NHS Economic Evaluation Database, and also the Cochrane Methodology Register. The ACP Journal Club is also an example of this. Unfiltered information could even include randomized controlled trials. Again, you have to filter through the questions that we've mentioned and through the grading system in terms of the bias of the trial and how the study design was set up. Cohort studies may be found on PubMed, as well as case-controlled studies. And then guidelines can be found in the National Guideline Clearinghouse, and other animal in vitro research, again, could be sourced from PubMed. These are just some of the corresponding databases to the hierarchy of studies. However, there's a proposed new pyramid. So traditionally we had the pyramid, as we discussed, from systematic reviews and meta-analyses at the top, going all the way down to expert opinions or case series. And the proposed new evidence-based pyramid looks at something different. It restructures or redesigns the pyramid. It actually chops off the systematic review from the top of the pyramid and uses this as a lens through which the evidence is viewed. There is also an acknowledgement that there can be overlap between the different study designs, as you can see in the wavy format. So what about qualitative research? Again, we have to rely on evidence that best meets the different problems that the clinician experiences in daily practice. So this may include a different format of a study design, and this could include qualitative health research. Adopting this approach, qualitative methods emerge as further points of reference, and this is with regards to contextual comprehension and contextual feasibility. It offers a more complex view of the way in which problems can be tackled. And this is important because it allows clinicians to have available a wider range of studies of different types that may respond to different types of questions. And this is necessary to build a holistic way of the different solutions that can be shared with the patient. So I've mentioned bias a few times as we're going through the pyramid, the grading method that's used to grade studies from high to low, and using the ABC system and the numbering system. And I'd like to share the quote by Ben Goldacre here, where he says, "Positive findings are usually twice as likely to be published as negative findings." This is a huge source of bias in research, and he calls this a cancer at the core of evidence-based medicine. So we have to be aware of this when we are looking for research. Studies tend to be published that have a positive result more often, or twice as often, as those that have a negative result. And this in itself brings in a form of bias in what we're looking at. So what about evidence-based medicine versus evidence-based practice? So evidence-based practice involves basing clinical decision and practice on the best available evidence found in practice. So what is this best evidence available? Again, the hierarchy of evidence is fundamental to the practice of evidence-based practice, and it attempts to answer this question. However, the usual hierarchy of evidence allows a top-down approach, whereas the evidence-based practice approach allows a different view to be taken to identify the best evidence according to which is most applicable to the context of practice. This can also be based on practice experience and what has been seen to be working in practice. There are key steps involved in this. First of all, again, we have to formulate the question. We have to search for the evidence, so it follows a very similar structure to evidence-based medicine. You have to appraise the evidence and use the same tools that you used before for evidence-based medicine. You can apply the evidence as was discussed previously. And most importantly, you have to assess the experience within your own practice setting and translate this into clinically and laboratory practice. So is this actually working within my setting? And that is one of the key differences. So this brings us to the point of integrating clinical reasoning and critical appraisal into clinical practice. In this complex process, an important role is played by the scientific evidence that the clinician has at their disposal to complete the clinical picture and reinforce their ideas on individual cases. In this sub-process, clinicians adopt the evidence-based practice approach. The evidence-based practice must be considered as a continuous and intertwined sub-process part of the overall critical reasoning and critical appraisal process within the complex process of the existing critical appraisal framework. It uses the other two sub-processes, clinical experience and clinical context. Despite appeals for medicine to be evidence-based and scientific, clinical reasoning does not conform to the conventional criteria of scientific methodology. However, we have to find a framework to incorporate our clinical experience within the traditional framework, or what is known as the scientific framework. So critical appraisal is essential to combat information overload and how to sift through all the many papers that you can find when you do a Google search. Identify papers that are clinically relevant. And this is important for continuing professional development, CPD points, so for keeping up your portfolio, but also it is a requirement for the evidence-based medicine component of many membership exams and your own ongoing professional development. The key components of evidence-based laboratory medicine in comparison, if we now think through what we've looked at evidence-based medicine, is determining the efficacy of diagnostic tests. This is broadening the view of analytical sensitivity and specificity alone. It's posing the appropriate question again and finding the correct evidence that corresponds with the question. You can still use the pyramid of evaluation for the efficacy of a new test or treatment that we have just gone through. And it is important when you are trying to identify the technical quality of the test, the diagnostic accuracy, the change in diagnostic thinking or patient management, or changing patient outcomes ultimately, and also when you are considering societal costs and benefits, which is a question that's important in the laboratory context. So despite the strength of an evidence-based medicine approach, many evidence-based decisions do not translate to diagnostic testing. Monitoring treatment using diagnostic tests requires an understanding of other variations of these tests in patient populations. This is essential for laboratories and clinicians in order to make accurate conclusions and decisions based on our results that we receive in the laboratory. So the practice of high-value evidence-based laboratory medicine is quite complex, but navigatable. So I'd like to talk you through this and give you an overview and a framework that you can use in your setting in the laboratory. And this is the framework that can be used in a routine practice, routine laboratory. So evidence-based laboratory medicine can be used to decommission an old test or another part of the service that's no longer serving the laboratory. It can also help identify where a new test will impact the care pathway. It can also be used to respond to inquiries from clinicians and carers, so understanding what your customers' needs are, what tests to use and how to use them, the meaning of the results that you receive, and what action to take on the receipt of these results. It can be used for performance management or quality improvement of tests by audits or introducing new tests. And introducing a new test is, of course, always a big challenge in the laboratory. This is usually in response to our client's needs, the clinician requesting a new test, new guidelines coming out, the manager wanting business case implementation program, or policymakers changing new screening programs, for example. And then research and development, which is usually about changing the model of delivery of care and leading service transformation. So introducing the principles of evidence-based laboratory medicine into routine practice can be done with the help of a tool. It's usually based on the clinical policy problem and by introducing something called the A5 cycle. This is based on 5 A's. Ask the right question, acquire evidence, appraise the evidence, apply the evidence, and assess the evidence. And for many of you in the laboratory setting, this rings very similar to an audit cycle. Asking the correct question identifies the problem and whether the clinical needs are met or unmet. And asking the wrong question, not asking at all, invalidates subsequent steps or evidence that you might find. Next, acquiring the evidence, you have to find strong evidence. And finding new evidence, you can use searches that we've discussed for finding new information, and also the hierarchy of evidence and the grading system that we have also discussed. The appraisal of the evidence is important. We've gone through the process of critical appraisal, and it's also described as the process of carefully and systematically examining research. So this is just a reminder that it has to be careful and systematically done. Again, reminding you of the questions to be asked. Is the question clear? Are the results valid? What are the results? Are they applicable to my patient population? External validity is very important. When you apply the evidence, this relates to external validity and highlights the importance of asking the right question, because you have to match the evidence to the problem. If you recall, I mentioned the PICO framework that you can use, which stands for P, does the evidence apply to the patient or population, the setting? I, does my method have the required performance, and am I using the correct cutoff values? C, is the current standard of care similar to control groups? Another C, what is the change in practice required? And O, what were the outcomes shown in evidence? And are they appropriate and achievable, especially in my setting? Then you have to assess, and this constitutes two basic elements, including the learning experience and applying the principles of evidence-based laboratory medicine in daily practice. How do I apply this when evaluating new laboratory tests, you may ask? So apart from your traditional analytical specifications, the outcomes of a new test should be clearly defined and evaluated. Harmonization and risk management policies are very important here. There's emphasis on equivalent test results that can be traced back to the reference material. The Test Evaluation Working Group of the European Federation of Clinical Chemistry and Laboratory Medicine proposes a 14-item checklist arranged in the following 4 domains for this process. Firstly, identify the clinical management problem and desired outcome. Secondly, verify unmet need and existing solution. Thirdly, validate the intended use and how the biomarker contributes to the solution, or test in this case. Assessing the feasibility of the new biomarker or new test to influence the clinical practice and health outcomes of the patient is the final step. You can use the grade approach, which we've discussed, and this gives you judgments and direction about assessing the link between accuracy and evaluated outcomes, and also gives you aims that you can achieve in terms of moving from evidence to recommendations in practice. You can use the evidence to decision framework approach, which is a structured approach in formulating the question or making an assessment and helping you to determine the certainty of evidence. Analytical validation and clinical validation are distinct processes, as we know, in terms of a biomarker validation process. However, the two processes are interdependent. A reliable test should be used to measure the biomarker before the biomarker is implemented or introduced into the laboratory, and there are specific processes to follow. The rigor of the analytical and clinical validation performed for biomarker qualification should support the utility of the proposed test. A listing of qualified biomarkers with the FDA reviews describing the evidence leading to their qualification, and this can be found on the FDA Biomarker Qualification Program website and might be helpful to review to see how this is done. This flowchart provides an idea of the key steps for qualification of a biomarker for use in the laboratory. The need of the biomarker should be identified, as well as the context of use, and of course the benefit and risk to the patient. And finally, this informs the type of level of evidence needed to support the qualification and the evidence that supports the qualification. The R&D process in the laboratory for establishing new biomarkers is thus very complex, as you can see, and it is also based on certain factors that need to be taken into consideration other than the grading, critical appraisal, needs assessment, risk, and so forth that we have discussed. So there are a few logistical considerations that are related to the laboratory. There are funding arrangements that have to be identified, and also the involvement of diagnostic companies in making biomarkers available on an automated analyzer may be needed. So collaboration with industry and manufacturers. And the data obtained should be appropriate for use as a component of the package required for qualification of the biomarker. This example here is given for the FDA approval or qualification of a biomarker. And there are various different regulatory bodies that are used that usually approve a biomarker, and these can vary from country to country. The main ones are the European, the EMA, the FDA in America, and in the African context, there are different ones as well. And these regulations should be followed as per local need and specification, and also the flowcharts that they have. So using this example with the FDA, if the biomarker qualification is the intent of the application, applicants are encouraged to collect the clinical data needed to apply to the FDA for the qualification. Researchers are encouraged to initiate the qualification process with the FDA prior to submitting an application. So this is even done before you do an application for funding opportunity or announcement. And the reason is to hear whether this would even be considered or whether the FDA can do a pre-qualification. This can save you and partners a lot of time and paperwork in the long term. Then you would plan to obtain advice from the FDA on the development of the biomarker. This should be included in the application. And the FDA Drug Development Tools Biomarker Qualification Program contains a lot of details, and here's a link to refer to. I encourage you to look at this to develop an idea of how biomarkers and new tests are developed. And then finally, after having gone through all of this in terms of evidence-based medicine, critical appraisal of papers, having to apply evidence-based medicine in the laboratory context, understanding the hierarchy of evidence and grading of evidence, and research and development in the laboratory context, the question is, how does this apply to me beyond a CPD program? What else can I do with this? There is a lot, and I'd like you to become aware of this, because the laboratory knowledge that many of you have, the experience that you have, may be very valuable in these settings and needed. So we can divide it into the academic roles and industry roles, or pharmaceutical roles. With academic roles, you can be a chemical pathologist that can manage biochemistry laboratories that guide sample analyses or use samples. You can be involved in the interpretation of challenging and unusual test results. Quality assurance roles, you can work with patients and clinicians that experience metabolic disturbances and require specialist services. And you can also become involved in research, clinical trial work, or even be a principal or PI investigator in research that is being done. This involves publication work, funding and grant applications, and has a whole list of skills required for that as well. But as a baseline, your skills within the laboratory setting and knowledge is invaluable to this. So if you have an interest in that, these are the specialist subcategories that you can apply for. And even within your current setting, there might be roles available for this. In terms of industry or the pharmaceutical setting, you can work in government or private hospitals, medical laboratories, pathology laboratories or clinics with similar roles and needs as we've discussed on the academic side on the left. You can be involved in medical instrument manufacturing facilities that require the input of your knowledge, and also in management or leadership roles. This could be in public policy and government positions or leadership positions, where, again, your knowledge and frontline work and experience is required. So finally, I'd like to thank you for listening to this presentation and going through the journey of evidence-based medicine in the laboratory context and critically appraising the research that you find. And I hope this contributes to your practice, gives you more guidance, and also assists you on your career path forward as chemical pathologists and clinically laboratory staff. Thank you.